Hi, today I'm going to talk through the single award science. This is Unit 4, which is the practical unit, and it is Booklet B, and I'm going to look first at the foundation tier. So this video will be the foundation tier, the next one will be higher tier. So it's 1 hour long, and the total for the paper is 70 marks. And it has 3 sections. So section A is the biology section. So the first question talks about a student setting up the investigation below to test if light is needed for photosynthesis. So one leaf was covered with aluminium foil as shown, and we can see that here with a T-shape cut out of the foil, and then parts are labelled A, B and C on that leaf. It says the plant was left in a bright light for 48 hours. The leaf was then removed and tested for starch using iodine. Complete the table below. So looking at the table, we have the parts of the leaf A, B and C, which were up here. The colour after starch test, and then whether starch was present or absent. So it tells you that A was black, so starch was present, and A is this bit here, so that would have been exposed to the light. Then B is this bit which would have been under the tin foil, so it should have stayed a yellow-red colour, and starch is absent. And then in the cutout here of the T, that's part C, it would have gone black because starch was present. So 2 marks for those two being correct. Part B, complete the following sentences. Choose from bright, dark, chloroplasts, starch and iodine. So before the plant was left in bright light as described above, the plant was left in a dark cupboard for 2 days. This was to ensure that the plant leaves contained no starch before the investigation began. You may remember that's called destarching the plant. So 2 marks, 1 for each word correct. Then when investigating if starch is present in leaves, it's important that the chlorophyll is removed first. So suggest why. You have 3 to choose from, and you can see there I've circled B for the correct answer. But it's not because chlorophyll is toxic. I mean, if chlorophyll was toxic, we couldn't eat green plants. B is it would be difficult to see the iodine if the leaf was green, and C is the chlorophyll would stay in your hands. So it is B, it would be difficult to see the iodine if the leaf was green. Some very dark leaves, imagine a holly leaf, they're a really dark colour and it's hard to see the iodine then change, or the iodine when it was added to it. So part 2, the diagram shows how chlorophyll is removed from a leaf. Complete the diagram by adding the labels for A and B. So you see here there is a tripod and Bunsen burner, and on top of that we have a beaker, and then there's another beaker inside that. So the liquid here in A is ethanol, and in B then is just it's a water bath, or you could just say water. Now the X there means that the Bunsen burner is switched off. We know we do that because in the next question, explain why the Bunsen burner is turned off. It's because ethanol is flammable. Then explain fully why the leaf is dipped into hot water after the stage shown in the diagram on the previous stage. So it's dipped in hot water to soften it, and because when it's been in the ethanol, boiling away there in the ethanol, it becomes very crispy and brittle. So you dip it in the hot water and it softens it so that it can lie flat on your white tile. Okay, so 2 marks there. The first mark for softening it, and the second one so that it lies flat. Now, I'm sure if you didn't say on the white tile, you know, if you said some other equivalent, that would be fine. Question 2 is about how you would make a temporary mount of plant cells for observation under the microscope. So it says in your answer, you should describe how to prepare a slide containing plant cells, for example, onion cells, and how to focus the microscope so that the cells can be seen in detail. I'm sure you've all done this, making a slide from onion cells, probably in the junior school as well as at GCSE here. So the things to make note of is how to prepare the slide, and it gives you the example of an onion slide or an onion cell, but you could do something different, and then how to focus the microscope. Okay, so this is a 6 mark question, and it includes your communication skills and use of specialist scientific terms. So it's going to be one of these where there are marking points, and these are the points they're looking for. So the first bit is talking about, removing a section of epidermal tissue from an onion. So that's the little layer the like of sort of skin in between the layers of the onion. So if you're talking about removing a little bit of that and then placing it on a slide in water or in iodine, and then putting a cover slip on the top, then putting the slide on the stage of the microscope, then moving the lens or the stage away from each other, then focusing at low power first and then focusing at higher power. So you'll see there 1, 2, 3, 4, 5, 6, 7 different points. If you're making 6 or 7 of those points, that puts you in the 5 to 6 bracket. And if you're answering in sentences with full stops, and you're using good use of scientific terms, you're going to get into the 6 marks there. If you only had between 3 and 5 of those points and good communication skills, you would be in the 3 to 4 bracket and get 4 marks. And if you only had 1 or 2 points, you'd be in the 1 to 2 mark bracket. And assuming you had good communication skills, you would get your 2 marks. If you made no points, you got no marks. The next part of this one, the diagram shows an onion cell that was observed by one student. Other surrounding cells are not included. On the diagram above, label the parts X and Y, and you can see I've done that already. So X is the cell wall and Y is the cell. Y is not the cell membrane. Y is the nucleus. Just shows you I must have been on autopilot doing that one there. Apologies. Now, many plant cells, next part 2, many plant cells also contain chloroplasts. The onion cell in the diagram does not. Suggest why. Well, it's because onions grow underground, and chloroplasts are only in the cells that are in the light. So 2 marks there, 1 for each of those points. Now I have another part of this question. The students counted the number of cells they could see in their field of view at high power. The results are shown in the table below, and you see here different students, A, B, C, D and E, and the number of cells that they have seen are there. Calculate the average number of cells counted by the students. So what we're doing there is taking each of these numbers, 21, 19, 11, 20, 24, and adding them together, and we get 95. And then there are 5 students, A, B, C, D and E. So 95 divided by 5, which gives you 19. So 2 marking points there. 95 divided by 5 is the first mark, and then the correct answer of 19 is the second mark. And finally, about cells, use the information provided to suggest one reason for the difference between the students' results. So if we go back and have a look here, the students have, as you can see, between 11 and 24 as their results. So you might suggest that because the cells are different sizes, so students might have different numbers. Or you might have said that students may have counted the cell parts. If you look here, 1, 2, 3, 4, 5, 6, 7, if you counted those cell parts, that could take you up to 24, whereas if you were just counting the single cells that you could see all of, you see I'm putting a dot in them here, 6, 7, 8, 9, 10, so that you would get much fewer numbers if you just counted the whole cells. Okay, that takes us into section B, and this is the chemistry section. So it talks about the reactivity of metals and says they can be compared by observing how they react with water or acid. Name 2 safety precautions that should be taken when observing the reaction between a very reactive metal and water. So the main ones I would say here are wearing safety goggles, using a safety screen, or using small amounts of metal. So any 2 of those will get you your 2 marks. Then name the gas produced when metals react with water or acid. Your choices here, you're given 3, carbon dioxide, oxygen or hydrogen. So the answer of course is hydrogen. B, the table below describes some observations when metals react with water. So we have here sodium and potassium, copper and calcium. Those are the 4 metals, and then the observations. We see sodium moves on the water surface and disappears. Potassium burns with a lilac flame, moves quickly and disappears quickly. Keep saying quickly in there. copper sinks in the water and calcium sinks, starts to rise and fall, producing bubbles. So put them in order of reactivity, starting with the most reactive first. So potassium is first, then sodium, then calcium, and then copper. All of those correct would be 2 marks. Only any 2 of them in the correct order would be just 1 mark. So potassium, because it says quickly, we know that it's reacting faster than the sodium. The calcium reacts after a period of time, and copper just sinks. So it has to be the order. Suggest one thing that had to be done to make sure the comparisons were valid. So I would have said the most obvious is the same amount of metal in each time. And then how would the reaction of metals change if at all if acid was used instead of water? So if it's acid, that means it's going to react much faster. So out of the answers that we have, less reactive, more reactive, and no change, we're going to choose more reactive. Next talks about beer making. So this is probably not something you would have done in school. But it says in beer making, the acidity of the brew needs to be tested throughout the brewing process. There are 2 important steps in the determination of acidity. Filter a sample of the brew into a test tube. This is step 1. This removes any solid material. So this is obviously going to be looking at something to do with filtration and separating techniques, which we would have talked about. And then step 2 is testing the filtrate with an indicator or a pH probe. So testing the pH. So draw a labeled diagram of the setup to complete step 1. So step 1 was filtering into a test tube. So we draw the test tube and then filter funnel and filter paper inside. So for having all of those things drawn and labelled correctly, so drawn was 1 mark for each thing, and then labelled correctly for all of them was another mark. Okay, so if you just draw the diagram without labels, you're only getting 3 out of the 4 marks available. Then B, an appropriate indicator is needed to test the filtrate in step 2. The indicator needs to be able to detect pH across a wide range of pH values. The table below shows a number of indicators and their colours in a range of pH values. So quite an intimidating looking table there, to be fair. But you see we have red and blue litmus paper, universal, and then a thing called methyl red. And then the colours at different pHs. So R is red, B is blue, O is orange, Y is yellow, G is green, I is indigo, and V is violet. So you see if we look at red litmus here to begin with, we've got a whole lot of red here, and then just blue, so it's really only 2 colours. With blue litmus, it's the same, we've got the same colours. With methyl red, we've got red here, and then we have yellow. Okay, but with the universal, you see it goes red, orange, yellow, green, blue, indigo, violet, like a little rainbow. So choose an indicator from the table above and explain why you've selected this indicator. So the one that you would pick is universal, and the reason is it's the only one that can distinguish between a range of pHs. So if you want to, it says you need to be able to detect pH across a wide range. None of the others actually will fill that brief. So below is a diagram then which shows another way of measuring pH. We've got a pH probe in here and a solution, and you see it's attached up there, and we're getting a reading of 3.2 on a screen. So give 2 advantages of using a pH probe instead of an indicator solution to measure pH. Now note that it's saying an indicator solution and not indicator paper. So be careful that you're not mentioning paper in your answer at all. Okay, so the first thing I've said is it can be used to show pH over time, so you can monitor the change in pH using a pH probe. And the second thing is that the value can be given to 1 decimal place. And another way you could say that, I suppose, is it doesn't depend on the judgment of the user. So if you're looking at an indicator solution, you have to compare it to a colour chart, and then it's a bit subjective as to what colour it actually is. Right, now see, there's a whole lot of reading in this one, and quite often you get quite a lot of reading in a question. It's really important to stop and read it and not just skip to the very last bit. where it says here you should start by drawing a labelled diagram, and then you'd be hooking back through the question to see what the diagram was of. So it's important you read it all the same way you would read a comprehension in English before you dive in and start answering the questions. So it says when an alkali is added to an acid, a reaction referred to as neutralization occurs. In this reaction, an acid and an alkali form a salt and water. When a person is suffering from indigestion, hydrochloric acid is collecting in their stomach. Indigestion remedies contain an alkali which neutralizes the acid, leaving the person feeling better. So a student wants to determine what volume of alkali would be required in a bottle of indigestion remedy in order to neutralize the typical concentration of stomach acid found in the stomach. The student is provided with a suitable concentration of hydrochloric acid. So describe how the student would carry out the investigation to find out how much alkali would be needed to neutralise the hydrochloric acid. So the important bit here is how much alkali would be needed to neutralise hydrochloric acid. A burette attached to a stand, beakers and graduated cylinders are provided. You should start by drawing a labelled diagram. So telling you have a burette here. So in the diagram, first of all, I've drawn and labelled the burette, and then it says you also have beakers and graduated cylinders. So you've got a beaker with some acid in it, and that is for the second mark. So only 2 marks for that diagram, okay? And then describing what you would do. So you're looking to neutralise the acid, so you're adding the alkali to the acid for 1 mark, recording the pH as you do that for your second mark, and then you say that neutralisation happens at pH 7, or some other way of expressing that you know that the reaction then has finished at that stage. So there are 5 marks in total for that question. There's a little bit of space on the next page to write that on as well, so I assume when this was printed these pages were face to face, because that way you wouldn't have to flip backwards and forwards across the page to see it. So 2 marks for the diagram and then 3 for the description here, and you see there the 5 marks there. Now then it says you've got a graph plotted by the student when they had completed collecting their evidence. Complete the labelling of the y-axis on the graph above. So if you've got a scale that goes from 0 to 14 and you're talking about neutralisation, you know that that is pH. That's 1 mark. Then the volume of alkali should be added, what volume of alkali should be added to the bottle of indigestion remedy, and explain your answer. So you see here the graph goes up slowly and then it goes straight up. That's where neutralisation is happening, and then it increases again slowly. So you want this, the value here at the portion of the graph where it's going straight up, where it has neutralised, and when you read that off the graph, it's 100 cubic centimetres. Your explanation is that's enough that is needed to neutralise the hydrochloric acid. So 3 marks there, 1 for the volume, 1 for saying neutralise, and 1 for saying the hydrochloric acid. Now we're into section C, which is talking about physics stuff. So in this question, the diagram shows how the speed of reaction can be investigated by 2 students. And you see here we've got student A and student B and a ruler. So it says, using the information provided, describe fully how this equipment can be used to find which student A or B has the faster reactions. I'm sure you remember doing this experiment yourselves, probably with a bigger ruler, maybe a metre ruler. But what you need to say here is that student A is holding the ruler and dropping it, and then B is catching the ruler as fast as possible. And then you're recording where the hand is on the ruler, which indicates the reaction time. And then for the 4th mark, you're swapping and repeating the process for B so that you can compare the reaction times of A and B. So those are the 4 main points you'd need to make. Then state one way that these students can make sure their results are reliable. So if you ever see the word reliable in a question about experiments, you know that they are looking for you to say that you have to repeat it. So we repeat results for reliability. So 1 mark just for saying that. And it says reaction times can also be compared digitally by individuals pressing a buzzer when they see a particular shape flash on a screen. So 2 students had several attempts at measuring their reaction time, and their results are shown in the table below. So we have M and J, and you can see here they have 3 attempts each, and it says, first of all, calculate the average reaction time of student M. So what you need to do, there are 3 attempts. So we're going to add the 3 values together, 0.47, 0.38 and 0.36, which gives us 1.21. And when you divide that by 3, it's 0.403 is the average reaction time of student M. Now you could give that just to 2 decimal places, which would be 0.40. Summarise the results then in the table above. So when you look at the table, if we look at M, first of all, they're going 0.47, 0.38, 0.36, and J's results are higher each time, 0.67, 0.61, and 0.55. So the first thing you can say to summarise the results is that M has shorter reaction times than J. But you'll notice as well that as time goes on, 0.47, 0.38, 0.36, the times are getting smaller, and for J, 0.67, 0.61, 0.55, times are getting smaller there as well. So for both of them, they decrease. The reaction time decreases as they practice. And now I've got one about a little circuit. So the circuit below was connected by a student who was investigating the relationship between current and voltage for a length of resistance wire. Using the letters A and V, label the ammeter and the voltmeter in this circuit. So you need to make sure you get these the right way around. The ammeter would always be in series, and the voltmeter would be in parallel then with the resistance wire. So how would you describe the arrangement of the voltmeter in the circuit? So you see here, in comparison to the wire, it's in parallel. And then state the function of the variable resistor in this circuit. So that's it there. And its function is to change the current. Variable means changeable. Then it says add an arrow to the circuit to show the direction electrons flow around the circuit. So electrons flow from the negative towards the positive. So when you look at the battery, the longer bit of the battery is always the positive and the shorter bit is the negative. So here there are 2 cells together, and that helps you work out which way it's going. So the current is flowing from the negative towards the positive. So in this case, if you're doing it at the top there, from right to left. Now part B, the student recorded the values of current and voltage displayed on the meters in the table below. On the grid below, plot and draw a line graph of these results. So I've put the results in here, and we have, if you get all 5 points correct, that was 2 marks. Only 3 or 4, you got 1. And then a best fit line would be 1. Now, I've drawn in a line here. My feeling is it should be a curve. It says in the mark scheme best fit line, but I think a curve of best fit is probably appropriate there, so 1 mark for it. Then use your graph to find the current when the voltage is 0.5 volts. So what you do there is you draw your line across 0.5 volts, which is here, and then down there, so 1.5. And if you look at that a bit more closely, you'd be more accurate than me with that. Then state the trend of your graph. As the current increases, the voltage increases. So just 1 mark. If it was 2 marks, you'd always have to, give values in there when you're talking about your results. But just for 1 mark, just the trend. Then it says when the voltage across the bulb is 1.5 volts, the current through it is 4.5 amps. Use the equation resistance is voltage divided by current to calculate the resistance of the wire. So resistance is voltage divided by current. The voltage is 1.5, current is 4.5, so 1.5 divided by 4.5 is 0.33, and the unit ohms is given to you there. Then the final part of this paper says, using the information provided, state 2 things that were done to ensure the investigation was a fair test. So we're looking at then, let me just go back to this. So it was for a length of resistance wire. So you'd have to make sure that it was the same length of wire each time, the same battery being used, and also you could have said the same voltmeter or the same ammeter. So any 2 of those would have got you your 2 marks. So that's the end of practical paper B. It gives you an idea of the sort of things that might come up. I'm going to look at the higher tier paper as well, so you might want to look at it too.